was teasing when I said naked outlet, but I mean, it can be transferred that way. Okay, so any questions about Helicobacter pylori?
So let's just say then, for instance, my daughters, if we're sharing the same cup, now it's gone, it's been treated with medication. Would they carry anything?
They could carry it, sure, absolutely. And again, and not show any symptoms of having it. Like I said, many of the people that are infected with Helicobacter pylori don't show any symptoms. Okay, viral diseases of the upper digestive tract. Herpes simplex virus, herpes simplex 1. This is a widespread disease, transmissible with saliva, persists for life. Some estimates are as much as 90% of the population is infected with herpes simplex 1. Usually you get it in childhood, and you get formation of blisters, sores on the lips. We also know them as cold sores or fever blisters. And you get them, generally, the transmission, I don't know, I might be making this up, but the transmission a lot of times is going to be via your parents. You know, I think, because you think about it, parents are always kissing on you, and usually it's children that have been, I mean, you've been exposed usually as a child, and if parents have any kind of fever blister or cold sore, those are going to be producing very unlike mad, and even if they don't go and kiss you, I mean, if they're touching themselves, they can have it on their hands. And so a lot of times our exposure to this particular microbe is via our parents, because they've already been exposed to it. Yeah.
Are canker sores herpes?
I think that's something different, actually, but I'm not really sure. Yeah, I think the canker sore is more of a, I don't know. I think it's a irritation, isn't it? I don't know. I don't know if it's a.
So can you not get it if you can't be? It can't be transmitted unless someone has the sore itself.
Right. Good point. So the microbe is latent in the nerve cells, and so it's not going to be transmitted unless, of course, you've got visible sores, and then you've got lots of variant being released. So it is generally associated with the lips and the mouth, but you can find it in other parts of the body. It'll multiply in the epithelial cells, destroys the cells. The DNA is going to persist in the cells, but here's the key. The DNA is non-replicating. So it's in the nerve cells, but it's not going to replicate and spread to other nerve cells. It's an late infection that manifests itself for any number of reasons. A lot of times stress can lead to the manifestation of it. Immunocompromised people maybe can express it. Diet changes sometimes.
Can it show because of heat?
I'm sorry?
In hot weather? Does it come out like cold sores and stuff like that?
Ultraviolet exposure can cause it to come out, yes. So I don't know about heat itself, but UV exposure certainly can cause it, and that would be happening in the summertime. So I suffer from herpes simplex 1, and you can generally feel it coming on. There's a little tingling that occurs when you feel the blister coming on, and I use this thing called Campho-Phenique. I don't know if you're familiar with it. There's other stuff out there. There's an over-the-market cream. I can't think of it now, that you can use as well. Abreva, that's it, yeah. Abreva that you can use. Just attach it. A lot of times, if you treat it when it starts to tingle, you can prevent the breakout from happening. At least that's been my experience. Any comments, any questions? Mumps, another viral infection. This viral infection then attacks glands. One of the major glands it attacks is the parotid glands. We don't see it so much in the United States now because of immunization, but we do still see it worldwide. There's a fever, there's a loss of appetite, headache and pain. Swelling of the parotid glands takes place. On some occasions, you can get the microbe moving to the membranes surrounding the brain and spinal cord, so you can get meningitis. Takes about 15 to 21 days after exposure. Exposure is by way of inhaled droplets of saliva. So it then enters into the bloodstream. It's going to reproduce in the upper respiratory tract. It's going to enter into the bloodstream. From there, it's going to make its way to the parotid glands or other glands too. The testes and the ovaries are also targets for this virus. It's a major problem for post-pubertal boys, or boys past puberty, because it can attack the testes and cause serious damage. The inflammatory response that accompanies this is what causes the swelling that we experience and the pain that you get from mumps. Humans are the only host, and the infection carries a long life immunity. So once you get it, if you survive it, then you'll be immune to it. We do have an attenuated vaccine. Any questions about mumps? Okay, well, let's move down then to the intestines, small intestines and large intestines. Just in general, the diseases that we see that infect the intestines, the conditions that accompany these diseases generally are diarrhea, loss of appetite, nausea, vomiting, fever, all these things that we would call gastroenteritis. Commonly the stomach flu. And there are a number of different things that can cause these conditions. Generally spread via fecal-oral route. One of the more serious conditions is dysentery. This is diarrhea that's going to have pus and blood in it. And as we've discussed earlier, we're talking about Vibrio cholera infections and stuff. In all cases almost, when you have some kind of diarrhea, vomiting illness going on, the best treatment is oral rehydration therapy. Be sure that you replace those fluids that you're losing, because that a lot of times is what leads to the more serious conditions, leads to the death. You just can't get those fluids replaced. A lot of people that die from Vibrio cholera do so because they become dehydrated. So there are three main species of bacteria that we find in the intestine. We'll find Vibrio species, I should say, that cause intestinal infections. There are a number of others that I won't mention here, but Vibrio species Vibrio cholera as an example. These microbes are generally salt tolerant. Campylobacter jejuni. This is a microbe that we commonly contract from fowl, so turkeys and chickens and ducks. And Enterobacteriaceae. These are the enterics now. These are the gram-negative rods, the E. coli, the Shigella, the Salmonella, the Yersinia pestis. So, pathogenesis can involve a number of mechanisms. As we've seen, some strains within a species can vary depending on the types of receptors that they might have, the cells that they target. Some microbes might employ more than one mechanism. So we've talked about some microbes that, like Vibrio cholera, just colonize the membranes and just produce a toxin. But there might be those that produce a toxin like Staphylococcus aureus does externally in food, but then also can invade tissues and cause damage by invading tissues. So there may be different mechanisms employed depending on the microbe and the conditions. And the other thing that we see, especially with this group of microbes, is a type 3 secretion system. A type 3 secretion system is a means of delivering a chemical or a protein or a gene product, if you will, to the target cell or the host cell. Essentially, there is a hypodermic needle, if you will, that is injected into the host cell, and then the gene product moves in through this needle. I want to focus on this group here for now, the Enterobacteriaceae, the enteric bacteria. The intestinal microbiota, but they're not just in the intestines. We can find them in water, we can find them in soil, decaying vegetables, and they can be pathogenic, they can be opportunistic. When we consider the pathogenicity of these microbes, a couple of things that we need to keep in mind here. All the members of Enterobacteriaceae have these three things here. They have a core polysaccharide, what we're talking about here then is the LPS, the lipopolysaccharide on the outer membrane. Okay, so they all have this. It's a common antigen now that's going to be shared by all cells. So our immune system then, if we can recognize this common antigen, we can then respond to all of these cells. That's where the toll-like receptors come in place. They're going to recognize this core polysaccharide here. There's an O polysaccharide. If you remember that O molecule sticking up there, we consider our gram-negative membrane. There was that O molecule, served as an identifier. So we see various antigenic epitopes with regards to the O polysaccharide. So while all of the strains have this, and all of them have these are very variable. Finally, you remember there was also the lipid A molecule that we found on these gram-negative membranes. When the lipid A molecule is released into the bloodstream, you get an inflammatory response, So it triggers fever, lets the body know that it's infected. And then we can use serological testing then to help us distinguish between different strains. Let's consider some of the virulence factors. A lot of these microbes will have a capsule or produce a capsule. And so that right away is going to interfere with phagocytosis. They have membrane and adhesin molecules. These are really just the pili that are extending out. And on the tips of the pili, we'll find adhesin molecules that allow them then to attach to specific cells. It really allows for them to attach tightly to these cells. And of course, that's the first step in infection. Many of these microbes produce some kind of exotoxin.